Good morning, students. So today's lecture is all about breeding procedures for quality. And in the breeding procedure for quality, we have already discussed introduction, certain quality traits that are because we are to discuss today breeding methods for quality, right? So one must be familiar with the quality traits that we have already discussed in the lecture that was delivered before. Even I made you all familiar with the sources that are required for having the quality traits. Sources also we have covered. Some of the breeding approaches we have covered. That was all about hybridization, interspecific hybridization, right? 3, 4 methods we have already discussed. Then we often discussed the important concentration, important notes that were to be kept in mind while going for the breeding for quality. And at the end, we often discuss the limitation. But I could not go in detail for this part. So today I will be discussing with you all genetic engineering, how quality can be improved as a result of genetic engineering. Okay, before I actually go to the modern approaches that is genetic engineering, there was one question from Santosh in my last lecture, and that was madam, what method is a significant out of backrows and pedigree method? There, on the basis of data description, I told you like we could go for the backrows method. But I would like to add one more option into it, like one should go for which method, like one should go for the backrows method or the pedigree method. And the result is its answer is very easy. One has to go for the backrows method in case one variety is agronomically unsuitable, that is undesirable, and the other is agronomically desirable. If you remember in the back rows, we only said one can go for we have to go for the back rows methodology only when we have one agronomically undesirable variety, but this is resistant, and on the other hand we have agronomically desirable, but it is susceptible, right? So this is the main reason why we need to go for the back rows method or pedigree. But one can go for the pedigree if, because if you people remember in the pedigree, I said both the varieties are agronomically suitable. One was having one was having the combination of one desirable character, and in the other variety there were many desirable characters combined together. But I told you about macros, like I said, because here we are not we are using this marker assisted breeding. So this was all based on the genetic results were all based on the genetic, and here we were not conducting any preliminary yield trials. So this was the reason I told you we could go, or we could out of this backrows is going to be the beneficial. My answer is still the same. But here just I added why we need to go for the backrows. So why we need to, although this question is little bit different from the one that was asked by the Santosh, but here I would again to inform you all, like in the backrows, one has to go for the backrows methodology. One has to go for the backrows technology only if there is one agronomically suitable variety that is desirable and other one is no more. Okay. And in the other case, one has to go for the pedigree if both the varieties are desirable. This was among breeding approaches, right, that we discussed in the last lecture. So I'm coming back to my today's lecture. We have already discussed the breeding approaches, and that is all based on the conventional methodology. But today we are going to discuss the approaches that are all based on the modern aspect. And here modern aspect is all about genetic engineering, where we can either modify the seed protein quality. See what we have. We have a character, and this character over here is regulating certain quality, and the quality we are talking about is all about protein quality, right? We are all talking about the protein quality. So how the protein character can be improved? Say suppose this is the gene that is this is the gene A that is actually controlling the protein quality. Very first, if we see this is no more giving you good result, we can modify it. Very first step is you go for its modification. Modification is further of two types that I let you know in brief later on. Second is if one second thing can be sometimes quality trait is shown in excess. In that particular case, we can suppress the endogenous system, and in third, we can go for the production of certain chemicals that itself lead to the improvement of the quality. So these three are the factors that we are going to discuss today in the topic that is breeding for quality, right? So before I go into the breeding for quality, because these all three methods are discussed in the genetic engineering, and as already number of times I have introduced you all with the concept genetic engineering. Again today I let you know genetic engineering is all about the introduction of a new gene. The new gene that is introduced is said to be transgene. New gene that is introduced is said to be transgene, and this is genetic. That means some changes are being done in the genetic content, but with the help of technology, and the technology is said to be recombinant DNA technology and genetic transformation, where genetic transformation concept stands for the intake of external foreign DNA that is said to be transformation, and recombinant DNA technology is the process whereby this DNA is transferred by inserting it into a vector which becomes recombinant, and later on it is transferred to the host cell. So genetic together is altogether the transfer of a new gene. That means wherever I say new gene, that means it is a transgene, and the transfer is taking place with the help of the technology, and that technology is further composed of two technologies. Very first is a recombinant, and another one is a genetic transformation. Why we need to go for using genetic engineering? Obviously because our today object is all about to study breeding processes for the quality. So we are looking after the improvement of quality traits like protein, oil qualities, but today I will be discussing only of about proteins. Sometimes when we go for the genetic engineering, it is not always we are going for the modification, we are going for the suppression. No. Sometimes there is often, as I discussed in the third methodology, that is biochemical production, as a result of integration of this transgene into the host cell, there seems to be the production of newer biochemicals, right? Or say proteins. Because we are all together to go for the improvement of quality, and that quality can be either by the result of modification, it can be due to the result of suppression, or it can be due to the production of certain biochemicals. So these three are the methodologies by which we can develop the, or we can improve the quality of the protein. Very first methodology is modification of seed protein quality, then suppression of endogenous genes, and then biochemical production, right? So here I go with very first method under genetic engineering, and that is modification of seed protein quality. Say when we discuss modification of seed protein quality, name itself discuss. That means here we are going, modification means what? Improvements. That means here we are going for the improvement of proteins, because these are the seed proteins. Then obviously these are the proteins which are stored in the seed. That means we are going for the modification of seed storage protein quality. But we are going for the modification through what aspect? We are going for the modification through modern approaches, and that modern approach is the genetic engineering, right? This objective, that means where seed storage protein quality is to be modified, it is to be improved. This particular objective in turn is grouped into the two broad categories, where very first one is how modification can be done. If I tell someone say if I tell some student like, see, you have this seed with you and you are to go for its improvement, right? This is the assignment that is given to you. Assignment is what? This is the seed and this seed is to be improved, right? How one can go for improvement of the seed? There are only two approaches for improving the seed. Either whatever gene is present within the seed that is controlling the quality, either you go for its modification, right? Either you go for its modification, and in the scenario where modification is not possible, why when you are capable of seeing like after modifying this gene for a number of times, it is still not giving the improved quality protein characteristic, then why to go for the modification? In case this strategy fails, otherwise it does not fail, but just giving an fake example, if it fails, then what is an alternative? Alternative is you you isolate a gene from the outside, and then this gene is transferred at this place. That means what is being done here? That means here is being done the introduction of a newer gene, and that introduction of a new gene, whatever gene that is introduced from the outside, it is said to be transgene, right? So here is second aspect is what? Introduction of an appropriate transgene. So these are the two broad categories by which one could go for the modification of seed protein quality. Modification means improvement. Improvement of what? Improvement of protein that is stored within the seed, right? Its quality is to be improved. And how improvement is done? It is to be done through genetic engineering. And genetic engineering is what? It is one of the modern approach. Now I will come to the very first category of modification of seed protein quality. That is, this is the one methodology of genetic engineering. And within the genetic engineering of modification of seed protein quality, I'm just going to discuss the very first parameter, that is introduction of an appropriate transgene. See, before actually I enter into the process, what is done here? Any new gene is isolated, but this new gene, again I repeat, it is called as transgene, because the method itself is the introduction of an appropriate transgene. Transgene means new gene. So obviously we need to go for the very first step is we need to go for the isolation. We need to go for the isolation of a new gene. Very important. This new gene must be appropriate. That means it goes for the improvement of, it actually goes for the improvement of protein quality. That means whatever new gene that is that is being isolated, it must encode storage protein. And what protein is to be encoded? The new this, if I say this is the newer gene that is being introduced, then this must, because we are looking after the our our very first objective is modification of Modification of seed storage protein. Now, since we are targeting the very first method where gene from the outside is being introduced, if gene is to be introduced from the outside, then this much must be clear into mind of you all. That means this particular gene, that means if we talk for this particular cell where this new gene is to be introduced, that means it is already deficient of this gene. It is no more having this gene. This is only the reason we are introducing the from outside, we are introducing that particular gene into the host, right? And it is introduced into the host so that the protein is actually the amino acid sequence, right? If any particular storage protein is no more inside the cell and it is to be introduced from the outside the cell, then and it is to be introduced from outside the cell, then ensure whenever it is introduced, it corrects the amino acid deficiency. That means the storage protein which was no more present into it, it is actually present there, right? So this is the process. But see, one problem that takes place is whenever any gene is introduced, it may be expressed anywhere. So it is very important, whatever gene is introduced, it is always seed specific. It is always linked to seed specific promoter, so that it is expressed only in the seed and it is no more expressed anywhere else within the plant. That means because in the seed we have sometimes from the seed we have plant also. So it is to be ensured whenever new transgene is introduced into the seed, it is always linked to the promoter that actually activates the seed specific, that is actually activating the contents within the seed, so that it is expressed only in the seed. So this is all about introduction of an appropriate transgene. But I will illustrate you this with the help of certain examples also. Very first example is about whistling. And this whistling, very important. And this whistling is the seed storage protein of pea. This is present in the pea. If we talk about its features, then this whistling protein is altogether having 7% lysine. It is having 7% lysine. But unfortunately, there is no amino acid that is made up of sulfur or that contains the sulfur. For example, I say methionine, cysteine. These are the sulfur containing amino acid. That means whistling is although rich in the lysine amino acid, but it is having very low amount of sulfur containing amino acid. And this thing is only to be improved as a result of say introduction of an appropriate transgene, right? This is the one example. Here it is a second example where we have a sunflower seed storage protein, and the sunflower seed storage protein is having and the sunflower is having a seed storage protein So here I was discussing about second methodology, second example under introduction of an appropriate transgene, where I was letting you all know that here we are talking about seed storage protein, and that seed storage protein is of sunflower. This seed storage protein is named as sunflower albumin 8. The protein that is found in sunflower is named as sunflower albumin 8, that is abbreviated as SFA8, right? And it is 2S albumin. Now, the one seed protein that we discussed earlier, that is viscellin, that was that was having low amount of sulfur containing amino acid, but it is having high amount of sulfur containing amino acid, and that was rich in 7% lysine, and this is rich in 23% of the methionine, and plus there is also the presence of sulfur containing amino acid, and one of the sulfur containing amino acid is cysteine, right? Now, what is to be done? We are to code for the gene that actually codes this protein. Now, the gene that codes SFA8, it is to be cloned, and this is to be expressed in alfalfa. From the sunflower, it is to be expressed in alfalfa. Just a minute. This is an example, and further I am illustrating that this gene, this sulfur albumin 8, sunflower albumin 8, that is S, that is sun, flower means F, and A means albumin and 8. This is although found in sunflower, but it has been cloned and expressed in alfalfa, then subterranean clover, and tall fescue to improve forage nutritional quality, right? But one thing, because whenever we talk for the quality, I told you while discussing the consideration for consideration, that is the points to be kept into mind while one goes for say while one was going for what we call as improvement of protein corrector. We must ensure like this, as we know, SFA8 is suspected to be allergenic. It is suspected to be allergenic. So it is not very attractive for food protein modification because it is having the qualities of allergy. So it is not to be introduced in any case when we are going for the food protein modification. Then another example I let you know, which is a study, and it has been found that in the recent study, 7S legume 7S legume seed storage protein, that is said to be beta physolene. This gene is driven by rice storage protein gene GT1 promoter, and later on it was transferred in rice. When it was transferred in the rice, then the modified rice, that is the transgenic rice, they often expressed 7S gene in their endosperm. Similarly, 11S legume in protein gene delivery by this also has also been transferred and expressed in rice endosperm. So this is all about introduction, where we isolated the gene and we have introduced into the host cell, and that host cell is later on allowing the particular modification to take place, right? And what is that modification? It is all about the improvement of protein quality. This was the one we have discussed till now, that is introduction of an appropriate gene. That was the very first objective of modification of seed protein quality. But right now we are discussing the second one, that is modification of endogenous gene. Modification. Say now this is the host, and within the host this is the gene. But this gene is not giving as good the result as were expected from this gene. Then what to do? Then what to do? Then one thing can only be done. This gene can be, very first step, this can be isolated from the cell, because we are talking for the modification. So after isolation, it is to be modified, and in the third step, it is to be again reintroduced back into the cell, right? and now this gene which was taken out, this gene is now modified gene. But you know, as we discussed in the last lecture also, any gene functionality is regulated by its particular promoter. While it was within the cell, its regulation was being controlled by a particular promoter. Its regulation was being controlled by a particular promoter. But when it is taken out, that is when once it is isolated, it is modified and then it is reintroduced back into the cell. What about its regulation? Regulation is to be done by its own promoter or some other specific promoter. That means the promoter that is specific to this modified gene is to be introduced. So in that case, I let you know, once it is isolated, it is modified and it is reintroduced, then its regulation, that means it is driven. Then this modified gene can be driven either by its own promoter, that means it can be driven either by this one only, which was already present within the host cell, or either its regulation can be done by a promoter that is no more present in the cell, but it is specific to this modified gene. So after the modification, its regulation can be either by the same, by its own promoter, or it can be by other promoters, but very important that promoter is again specific to this one only. If this promoter, other promoter is not specific to this one, then it will not work until unless it is allowed to be driven by certain promoter. So this is the modification of endogenous gene. That means that the system that is already present within the cell, it is modified. It is modified with the aspect that it gives the good protein quality, right? Now the next is one thing that is to be noticed while we are going for this modification of the endogenous gene. Modification, how modification is to be done? When you are going for the modification, that means you have certain sequence, and since you are going for the protein modification, then in the protein we used to have amino acid sequence. We used to have amino acid sequence, and you must have gone through genetics. And if you remember that within the, when we talk for the genetics, then in the genetics, say if this is the sequence, then here used to be the presence of bases, and say those bases are adenine, uracil and guanine. Then you know three bases code for one codon, which is read by the messenger RNA on the ribosome, and it gives rise to the protein, right? I won't go into detail mechanism of transcription. Right? So here what is happening out, I said that like while one goes for the modification, these three steps are to be followed and this is to be taken care of, right? But the gene sequence, how the gene sequence, this is a gene sequence, how it is to be modified? Modification can be done either by changing, that means by replacing one or few codons. This is the one codon, either by replacing one or few codons, or if you don't want to replace, what can be done? This can be removed, and instead of it, say not this one is removed, say this is removed, and instead of it another additional base can be added, so that it is additional base can be added. That means one more thing, when you are going for this replacing, either what we said, either we can replace one or few codon, or either we can insert one or few codon. But very important thing that is to be kept into mind again is whatever modification is done, it must be taken care of that reading frame of the gene is not changed. What is not to be changed? The reading frame of the gene. Like I told you, like here I told you, no, replacement of the codon is to be done. So one codon is to be changed with another codon, or another codon can be inserted. But say if the frame is reading frame is A, U, G, C, T, then this framing pattern should not be changed. This is to be taken care of while replacing or inserting the codon. So here is one of the example where modification of the endogenous system has been done. Like very first we are discussing about the protein that is prolamine storage protein. Prolamine storage protein that is called as zine in case of cereals. These are deficient in essential amino acid lysine and tryptophan. So how these can be improved? Either it can be improved by single lysine replacement, or it can be improved by double lysine replacement. This is done at the end terminal, and this can be done in between or within the peptide repeats. But whatever method is adopted, care is to be taken that reading frame is not changed, right? Because if reading frame is changed, then it will disturb the folding pattern and the stability of the encoded proteins. And we don't want in any scenario folding pattern and stability of the encoded protein to be affected. So this is the reason what is to be taken care of. Care is to be taken off so that correct reading frame of the gene is maintained. With this, I complete the very first method of genetic engineering, that is modification of the gene, which itself included two parameters, introduction or modification. Now we will talk about another approach, that is a suppression of endogenous gene. If we cannot modify, what we can do? We can suppress it. As the name itself is indicating, we are going for the suppression of the regulatory system that is present within the seed. So how suppression can be done? You don't want certain particular character to express itself, then what you could do for it? Either you eliminate it. What we can do is only the elimination of it. This is a particular gene which we want to suppress. That means we no more want it to express itself. Then how its expression can be regulated? How its expression can be controlled? It can be controlled by eliminating. Either you eliminate this specific gene or reduce its content to the amount so that it is no more expressible. There are basically four methodologies by which this endogenous gene expression level can be reduced, and these approaches are, very first is the antisense RNA, then is the ribosome, gene disruption, and by co-suppression. But out of these four approaches, the one that is very successful is the antisense RNA approach. So by going for this approach, we can very appropriately suppress the endogenous genes. But before you actually suppress the endogenous approach by antisense RNA approach, you must be familiar what is antisense strand, what is meant by the sense strand, and what all is there in this antisense RNA approach. See, before I actually enter into this antisense RNA approach, I would like to make you all familiar with the DNA. So this used to be the structure of DNA, where we say the DNA is double stranded. Where we say the DNA is double stranded. If I open its structure, then it is somehow, because we used to have two strands, one is extending from 3' to 5' direction, and another one extends from 5' to 3' direction. Here, kindly put arrow instead of this hyphen, right? 3' to 5' direction and 5' to 3' direction. The one that is extending from, very important it is, the one that is extending from 3' to RNA, 3' to 5' end is actually the one that is transcribed. Kindly listen it carefully, it is the important concept. In any gene, this is any gene, say this is any gene, right? This is any gene. In any gene, we have direction of the strands of say two strands, something like this. That means one strand is in the direction 3' to 5', and another one is in the direction 5' to 3'. Now, when we talk for the this strand that is 3' to 5', that is in the 3' to 5' orientation, but this orientation is in relation to its promoter, very important. Wherever its promoter is placed, from that particular promoter when seen, its direction is 3' to 5'. When this strand is transcribed, when this 3' to 5' strand in relation to its promoter is transcribed, then this 3' to 5' strand is called as antisense strand, very important. So what is the antisense strand? The strand that is in the orientation 3' to 5' and which when transcribed in relation to its promoter is called as antisense strand. Transcription matlab when from the this is the template, this will act as one template and it will result into the synthesis of mRNA. That means this is the, if I recognize red one as 3' to 5' strand in relation to its promoter when it is transcribed, say the sequence is T A, sequence of this 3' to 5' I am discussing, T A C, then C, then A, then A, right? Then the one that is providing the information, it is called as, because it is part of the DNA, one strand of the DNA, so it will be called as DNA, because it is providing the information, so it will be called as template. And since it is a strand, so it will be called as DNA template strand. Very important. When this when the DNA strand that lies in 3' to 5' orientation in relation to its promoter, when it is transcribed, this is said to be antisense strand, and when it is transcribed, that means it will result into the synthesis of mRNA. Then it will result into the synthesis of mRNA. Since this is the sequence over here, then mRNA is going to have opposite sequence, that is instead of T, it will be U. Instead of A, because mRNA, and when we talk for the RNA, then there is a replacement of, normally what happens, A binds with T, double bonded, A is double bonded to T. But when we talk for RNA, then A is bonded to uracil. This is the difference in DNA and RNA. Then is the C, instead of C here is going to be the G, and this is acting as one codon, because one codon is altogether the sum up of three bases. Then we have C, then again here it is going to be G, then we have A, here is going to be U because this is RNA, and again we are going to have U, and this is going to be the second codon, right? Now, every specific codon is synthesized for a particular amino acid. Say this is synthesizing methionine, and this is synthesizing. This is further linked to the another amino acid, and the amino acid that is coded by this sequence, it is said to be valine. In this way, I have represented only six bases. That means two codons, and two codons are synthesizing two amino acid, but it is further connected, right? So this combination of amino acid is called as protein. I just hope antisense strand is clear to all. This I transcribed and I show you the transcription process just because of the reason I wanted to know you all that the mRNA base sequence that is synthesized from DNA template strand is having the sequence opposite to that of the antisense strand. See, of course, it is having the complementary. The base sequence that is contained by this mRNA, it is opposite to this sequence. See, because complementary. Complementary means if here is T, here is gonna be A. But when A is there, then U, and so on. So this point is clear to all. That is any gene. What is antisense strand? That is in the any gene DNA strand in this orientation. When is transcribed, this strand is said to be antisense strand. And the mRNA that is synthesized as a result of it, it is complementary to the antisense strand. This is antisense strand and mRNA is complementary to antisense strand. These 2 points are very important because I need to discuss this process of antisense RNA approach. But before I actually discuss the process, you must be familiar with antisense strand and the sequence of mRNA base sequence. Now we have to talk about the remaining strand of the DNA. The remaining strand is this one, yellow one. This I can say this is the remaining strand of the DNA. Every DNA is composed of two strands. Very first is said to be antisense and another is said to be sense strand. We talked about antisense strand. Now the remaining strand is said to be sense strand. And you know, this sense strand is naturally complementary to the end because the two strands, always what happens, the two strands are complementary to each other. The bases of this strand are going to be complementary to this one, right? So obviously when we are talking for the remaining strand of the remaining DNA strand of the gene, this is said to be the sense strand. It is going to be complementary to the antisense strand. That means antisense strand, what was the base sequence of antisense strand? It was T. So here gonna to be A. Then we have A. One thing is to be noticed when we talk for the mRNA, then here T is replaced by the U. But when we are talking for the two strands of the DNA, then A is to be replaced by T. Right? Then next is C. So here is going to be G. Then again G, then again T, then again T. So sense strand is complementary to the antisense strand of the gene. Therefore, because it is complementary, this sense strand is complementary. Very important point I am going to discuss. Because this sense strand is complementary to the antisense strand, but there is one similarity between the mRNA that is produced from antisense strand and the normal sense strand. You could see the base sequence of sense strand is same as that of the mRNA that is produced by the antisense strand. That means we can conclude one thing. That means whatever heterogeneous population of RNA or mRNA is produced, it is we can consider these all the mRNA as the sense RNA. Antisense RNA and sense RNA. I told you this is the antisense RNA, and when antisense RNA is transcribed, it gives rise to mRNA. Now we talked about the second strand of the DNA, which is complementary to antisense. And what you visualized, this sense strand is same as that of mRNA that is produced from antisense strand. So instead going into complications of learning, this is mRNA, this is sense strand, we can simply say whatever mRNA these are produced, since they are having the same sequence as that of the sense RNA, so this heterogeneous population of mRNA can also be considered as a sense RNA, right? Now, one change I let you know. Here I said the sequence is going to be T. The sequence won't be T. The reason behind DNA, when we are discussing for the DNA and when we say for the single strand, then the single strand is what? The single strand is mRNA only, right? So here if the sequence was T, okay, here it is going to be A only, and here it is going to be U, and similarly here U. Now it should be more clear. Like see, both the sense strand and the mRNA have the same sequence. So this is the reason, even if I call it as a sense strand, it is not going to make any bigger difference. Even if I say, because if I kept two strands in front of you, here too you are capable of identifying the two strands. You can easily say this is ma'am, this is sense, because I have represented it with the yellow color, and here you can easily recognize like this is red because I have highlighted this one with the say red color. But if I don't differentiate the two with the colors, then it won't be possible for you to recognize it won't be possible for you to recognize which one is the sense and which one is the mRNA sequence. So a conclusion, what I could go for the conclusion, I could conclude like mRNA can be considered as a sense RNA, right? With this, I finish for the concept behind antisense RNA approach, because we are to use this antisense RNA for the suppression of the endogenous gene. So I made you all familiar with the concept that are related to the antisense RNA approach, where I introduced you all with the antisense term, antisense strand term. Then I introduced you all with the mRNA, right? Then I introduced with the sense strand, and then I concluded the antisense approach by saying all the mRNA, they can be considered as a sense RNA. Sense means that is going to give you certain beneficial product, right? So this is antisense RNA approach. Now, how antisense gene is to be constructed? How the antisense gene is to be constructed? When we talk for the construction of antisense gene, now antisense gene is to be constructed. That means this gene that codes this antisense fragment, it is to be constructed. It is to be prepared, right? How it can be prepared? It is produced by inverting the orientation of protein coding region of the gene in relation to its promoter. Say this is the DNA. This is, I hope just this is all clear to you all, because I'm going to give you another example now. Just a minute. I will be discussing the two side by side so that it becomes easy for you all to understand, right? Now see here what is represented. I just hope this image is clear to all. See here we have strand. I will be writing it over here, the sequence. Here we have two strands. And now I will be representing with one color only because I have taught you the concept behind it. Now the sequence, here the direction of the two strands of the DNA are, one is in the 3' to 5', and another one is in the direction 5' to 3'. You know 3' to 5' in relation to its promoter is said to be, this is said to be antisense RNA. Antisense, I'm writing it short form, you are not to write. This is said to be antisense RNA, right? And if this is antisense, we know opposite is going to be the sense strand. Now here is the location of the promoter, and this is the location of our sense strand. The sequence goes like this: 3' to 5 direction in relation to its promoter is said to be antisense strand, right? The sequence is somehow T, then A, then C. This is the sequence T, A, C, A, C, C, T, C, G. And this is the sequence that is shown at the 3' and at the 5' and this is a sequence. And obviously it is going to be complementary because the two strands could bind to each other only if they have complementary base sequences. So if it is G, it is going to be C and so on, right? And this is. See what I made you familiar in my last slide, like the one that is in the orientation 3' to 5' in relation to its promoter is the one that is going to be transcribed, and as a result it is going to give rise to the mRNA. mRNA means from T what is going to be synthesized A, but here this is mRNA, you know, the basis of the mRNA, it is altogether consisting of adenine, then it consists of uracil instead of thymine, then it has guanine and it is having cysteine. The difference is only in the presence of UOT. If U is present, then this represents the sequences of RNA, and if T is present, that means it is we are talking about DNA, right? But here transcription, as a result of transcription, mRNA is going to be synthesized. So A here is going to be replaced by U, and so goes the pattern, right? Last one I will be discussing from G what is synthesized to C. Here also it goes like A, A, G, G, A, G. So this is said to be mRNA, right? Now I will talk for how antisense gene is to be constructed. This is the one methodology that I discussed in the last, so I just hope it is clear to you all how mRNA is synthesized and how this is the sequence that is present on the mRNA. Now I am to discuss the construct of antisense gene. When I am to construct the antisense gene, then see, we said it is produced by inverting orientation of protein and coding region of gene in relation to its promoter. And what is going to synthesize gene? that is to be synthesized by antisense gene. Antisense strand is only going to synthesize mRNA, which is later on going to synthesize protein, specific protein. But here, while we go, this is okay. This is okay. I have told you this, and this is clear to you all. But when we are to construct antisense RNA, then for that, antisense RNA is constructed by inverting its position. Inverting its position means if we invert this position, this end will come to this end, this end will come to this end, right? If this is here, then opposite this side is going to be this because we have inverted, we have rotated its position, we have reverted its position, right? So if 3' is here, then opposite is going to be 5', and in this way, see, the whole sequence is also changed. This is how antisense gene is constructed. How it is constructed, very 1.1 word you need to remember. It is constructed by inverting the orientation. This is the coding reason. This coding reason is inverted. Now this is becoming this has become the coding reason, right? See, it is very simple. This is the 3' end has come here because you have inverted the position. You have reverted the position. So 3' is going to be on the upper side, and this is going to be on this side, and the opposite strand on this side, obviously. So if this is the sequence, here the sequence was A, so here the sequence is going to be, we have reverted the, we have inverted the position now. So the sequence is also going to be reversed. That means here very first will come G, then is going to come A, then again G, A. And similarly for this one, here we have C, then we have G, then we have A, then we have G, then we have G, then we have T, and then we have GTA, that is GTA, and the end is closed by labeling it as 5'. Similarly will come this sequence. This is the antisense gene construct, right? If you remember, we said, because mRNA, we said mRNA is having the same sequence as that of sense strand. So these mRNA can be considered as a sense strand, right? So here, this is the antisense, and when this is transcribed, when it is transcribed, because we have inverted, and now this is now antisense strand is the one that is in the orientation 3' to 5'. That means now this is acting as a antisense strand, right? So see, from G it is going to be C because RNA is synthesized. So from A it is going to be U, then C, U, and similarly this one. This is now antisense RNA. I will read from here and it will be clear to you all. Before I read, I wanted to explain you this concept so that it is easy to understand the points that are mentioned under construct. So very first is, and this antisense gene construct is produced by inverting. As a result, this this sense strand. Now the sense strand, again I repeat, this is the sense strand that is in the orientation 3' to 5'. It is not the antisense strand. It is the sense strand that is now in the orientation 3' to 5', obviously in reference to its promoter. And now it is the sense strand that is transcribed, and as I already said, it is not the antisense strand that is transcribed. If you could see, here was the antisense strand that was transcribed. But here the strand that is transcribed is said to be sense strand. I hope that the 2 points are clear. Again, I am letting you all know, kindly fit it into your minds. The normal antisense strand is not transcribed because this one is not transcribed here because its position has been inverted. And anti-strand is only the strand that is in the direction 3' to 5' with the relation to its promoter. But since we have inverted the position, this is no more going to go to give rise to the mRNA. Now this is going to give rise to the mRNA. Now the RNA that are produced by the antisense gene construct, RNA, now it will be transcribed. And after transcription, this is the strand. After transcription, it is going to give rise to Now it is to be inverted, right? Because here already we have 5 prime to 3 prime, and this is this one, right? This one, and this is this one. Okay, this is 5 prime to 3 prime, so obviously its position is going to be 3 prime in the direction 3 prime to 5 prime. So RNA that is produced by antisense gene construct, this RNA that is produced by antisense gene construct, it has the same sequence as the antisense strand. If you could see, this is having the same sequence as this one, only the difference is like instead of T we have U, and this is now called as antisense RNA, right? Now I will, this was how we constructed the antisense strand. Now what is the process of antisense gene? So once the antisense construct of the gene is present, once this antisense construct, this is present in the gene, because we have to suppress, right? So we have a gene, and a particular gene is not giving good results, so it is to be suppressed. This is what we are talking in for the second methodology under the modification, that is suppression of the antisense, suppression of endogenous gene. Here what is being done, when an antisense construct of the gene is present, transcription of two genes yield, obviously they are going to give rise to antisense and RNA. This is going to give rise to antisense and RNA. Since sense and antisense are complementary to each other, now this out of this, this will be acting as a sense strand because it is in the direction 5 prime to 3 prime, and it is in the direction 3 prime to 5 prime, right? So since their bases are opposite to each other, so they will pair. This is what their pairing has taken place, and once it is paired, this mRNA will no more be now available for the synthesis of proteins. And also, because mRNA they both have complementary sequence, and again they have converted into the double standard DNA. So this is now no more going to be available for the synthesis of proteins. But since here are two strands of the RNA, so it could be attacked and broken down by double stranded enzyme that is said to be RNase. The enzyme that is going to act upon the two strands of the RNA, that is said to be RNase enzyme. And finally, so these are the two conclusions as a result of pairing of these two strands. Finally, these events are known. When this much process will take place, it will lead to the methylation. It will lead to the methylation, and coding region of the normal gene will result in the gene sequencing, right? So this is the process of RNA approach. So I just hope this is clear to you all. So this was all about the construct and process of antisense gene. So now I will discuss with you all its applications. As a result of antisense RNA technology in tomato, this can be better explained by an example of flavor tomato, wherein tomato, due to the presence of an enzyme that is called as polygalactonase, this enzyme is responsible for degrading pectin, and pectin is one of the major component of food cell wall. When pectin will be broken, obviously when pectin will be broken, that means it is a component of cell wall. Cell wall used to be hard in the nature, and when there will be the lesser amount of the pectin or there will be the breakage of the pectin, it in turn will lead to the softening of fruit. When fruit will be softened, it will be deteriorated fast, and thus we can say that in the tomato, due to the presence of this enzyme, it leads to the softening of fruits, and as a result deterioration in the fruit quality, right? But on the other hand, we say if we have the tomato into which is introduced a foreign gene, that is we have transgenic tomatoes. Transgenic tomatoes are the one in which antisense construct is encoding for the enzyme polyglucuronase. And this transgene changes the expression of this enzyme. It is reducing, it is actually suppressing the level of this enzyme. As a result, there is a lesser softening of the fruit, and as a result, deterioration is also decreased. So this is the way how suppression is done, and how suppression is done, that is all together based on antisense RNA technology that I have told you, where actually antisense strand becomes dominant, it functions, and finally it is resulting into the non-methylation, and it is resulting into the degradation of mRNA, whatsoever were the conclusion, right? So this is the one example of flavor saver tomato. The tomato into which the newer gene is introduced is called as flavor saver tomato, and here suppression, again I repeat the word suppression of this enzyme is expression of this enzyme is suppressed, right? It has because when this next slide is in continuity with this transgenic tomatoes, if the amount of if the expression of say what we call a transgenic tomato, expression of enzyme within the transgenic tomato is expressed, then obviously it has improved the shelf life and quality of tomato, right? In addition, fruits they give improved flavor and superior juice viscosity. And these tomatoes were when sold out, when were marketed in USA, they were given the another name that is said to be McGregor, right? So this was all about antisense approach. Now we have the last and the third method under genetic engineering is all about biochemical production. Name itself is introducing, that means here we will go for the technology, we will go for the genetic engineering in a way that leads to the production of certain biochemicals, which may be toxins also, right? So here also, but no, see, normally cell is no more producing any biochemical. If it would be producing, then then very first only, at the very priority only, this would have improved, the technology would have improved. But since no biochemicals are being released, so you are again isolating a one particular gene, and that gene is inserted into the cell, that is, it is said to be transgene. This transgene is used to introduce many newer branches of the biosynthetic pathways, and as a result, valuable proteins can be generated. For example, thrombin inhibitor protein hirudin. This is the first example of this biochemical production, and it is produced by Napus, right? So what hirudin actually does, it actually encodes a synthetic gene, and this is expressed in fusion with oil baby protein oilacine that helps in its purification. What happens when seed tissue is extracted with water and later on when it is centrifuged, then what happens, because it is used for the purification, so this fusion floats on the surface and can be easily separated from the rest of the seed proteins. So what we are going for, we are utilizing hirudin, we are using for the purification of it for the purification of oilacine, right? How it is purified? Because it is forming a fusion with hirudine, and hirudine actually forms a fusion with the oilsin, and due to its fusion, it actually floats on the surface. Uh, because of the binding of the oilsin with the hirudine, it is floating onto the surface, and because rest of the proteins are no more binded to the oilsin, they are not floating and they are just present on the surface of the, say, on the bottom of all the content, right? So this one will be floating onto the surface and can be easily separated, and in this way it can be purified. So this is what purpose of the hirudine and how it helps in the purification. This is a flowchart for that. So actually here I am letting you know that certain biochemicals when produced within the cell, they further help in the purification of the particular say particular content. Later on, when you have separated it out, the bond can be cleaved by the enzyme that is said to be protease, because protein is there. So breakage will be with the help of enzyme protease, right? And some other, this is one successful example of hirudine, where hirudine is expressed, right? These are the another examples like gus, then avidin protein, right? These are in case of the maize, and these reagents are marketed, means the reagents which are required for its purification and all, they are marketed by Sigma in USA. So this was all from my side. So the today's topic was all dependent upon modern aspect of breeding for the improvement of quality in terms of protein, right? Where I have already discussed with you all in my previous lecture about the breeding conventional technologies. And today I discussed with you all about the technologies that are all under the modern approaches. And it is all about genetic engineering, where we discuss the three parameters. Very first is the modification of the gene, second is a suppression, and third one I made you all familiar is all about biochemical production. So with this I complete the lecture from my side. uh, Thank you, stay home and stay blessed.